So, in your lab, I hope that you observed osmosis, and we're going to describe that with a little picture and some words. So please follow along. We start with our beaker down here, and we have our egg sitting in it. That's not a very good egg, but you get the idea. Water's in the beaker, and remember, food coloring is in the straw. So what happened? What did you see? Yes. So the food coloring went up, right, over time? It increased. Why did that happen? Any ideas? Sam?
It wanted to spread out.
What wanted to spread out? What are we talking about? The proteins in the egg? Okay, so are there more proteins in the egg or in the water? The egg. That's important information. So there's a high concentration. Lots of proteins. And what else? What other molecule is a big component of eggs? In the yolk especially. Lipids, right. So if the proteins and lipids wanted to spread out, they would actually spread out into the water. Would that make the food coloring rise or go down? Go down. But you're right, something wants to spread out, and it does. Not the proteins and the lipids inside the egg. What is it? The water, exactly. So there's a high concentration of water. In fact, it's distilled. It's got everything removed except water. So it's 100%. Nothing else in it. You can't get any higher concentration than that. So the water will actually move into the egg. Because of the concentration difference. There's less water in the egg than there is outside in the beaker. That's what pushes the food coloring up. Sam does bring up an interesting point, though. Do the proteins and lipids, those are molecules, those are particles, like the smoke that we released in class, shouldn't those want to spread out? And they can't really want to because they're molecules. They don't have any cognition. But naturally, wouldn't the proteins and lipids diffuse? Yes. So what's preventing them from diffusing into the beaker? The membrane. So it's letting water in, but not letting proteins and lipids cross. Why is that, do you think? Will?
Because there's less water in the egg, so it wants to be more balanced. It wants to bring
That's true about the water. The water will diffuse until there's an equilibrium or a balance. The concentrations are equal on both sides. But the proteins and lipids, they will undergo the same phenomenon, except what's preventing them from doing so? The membrane. And why can't they fit through? The membrane does only let certain things in and out, but there's a difference between the molecules. This is only 3 atoms. Water is a small molecule. Remember, proteins and lipids are chains. They're polymers. They're very big, hundreds of atoms in some cases. So it turns out these are too big, too large to permeate the membrane. And like Chris said, cell membranes, they're actually designed this way to retain nutrients and get rid of waste. So cell membranes are semi-permeable, which means they let some things pass and they don't let other things pass. So our egg membrane is similar to an actual cell's membrane in that respect, and not everything can just pass through. Larger molecules have a harder time moving directly through the membrane, and also charged molecules. Hopefully this sheds some light or confirms what you have learned thus far in the lab. Are there any questions at this point? Number one, can the proteins in the egg white pass across the membrane? And support that with evidence. Remember, the egg is made mostly of proteins and lipids. Did that pass across the membrane into the water? No. Right. Why couldn't they pass through? Kyla already told us. Someone besides Kyla. Water could pass, but they couldn't. What's different about these molecules in water? They're larger. Lastly, what evidence did we have that the egg was not leaking out, or diffusing out, I should say, into the beaker? What would have happened if the egg was, the proteins were diffusing out? It would have went down. It certainly would have went up, right? Any questions on the first one? I will leave this up here, so if you can't write fast enough, I'll leave it up here and you can come back when you get time to work and scroll through it and revisit them. Remember, don't use my words, make them your own. The discussion is to help you establish your own ideas to support you, but you can't use my work for your lab report. Second question here. Explain your observation in terms of concentration, diffusion, passive transport, active transport, and osmosis. Let me start us out here. I know that there was a much higher concentration of water in the beaker, so let's start there. In the beaker, the concentration was higher of water. Because there was a concentration difference, what happens? Just like the smoke. It spread out. What do we call that? Sophia? Diffusion, right. And where did it diffuse across? What did it diffuse through? The membrane? And what is that entire thing called? Diffusion of water across the membrane? Yeah. We got a couple more words to use. We've used concentration, diffusion, and osmosis. Let's talk about this idea of passive transport. We talked about that in yesterday's bell work. What is that? Anyone remember? Or can read it from their notes? Jordan? The particles move with their own energy. The cell doesn't have to use energy, right? And how do they move? By diffusion. So if osmosis is diffusion, and diffusion doesn't take any energy, could you also say osmosis is passive transport? Yeah. And then this last idea we have to talk about is active transports, which is the opposite of passive. So can someone speak to that? What's active transport? Passive is when the particles move from high to low and it doesn't take energy. So active would be from other way, low to high. So you're actually pumping molecules into a space and making them more and more and more and more concentrated. Did we show that in the lab or not? Can the egg pump anything? No. We did not show active transport, which is molecules moving from low to high. This is a long question. This is the longest one. Number 3, this is based on your own work. So can anyone share if their hypothesis was right or wrong? How many people thought the liquid would go up? Let's just do a quick show of hands. How many thought it would go down? How many just didn't know? Okay? Whatever prediction you made, even if you didn't know and you just kind of had to guess more than you would have liked, make sure you tell if your data agrees or disagrees with it and refer to your data. If you said, we hypothesized that the liquid would go up, you should follow that up with, our lab confirmed this idea. It went up this much in this period of time. So refer to your data. Use your numbers in that question. Number 4. Is this an example of active or passive transport? We said that in number 2 already. The egg showed passive transport. Why? What evidence do we have? How did the molecules move? I've asked this about 4 times now. Probably an important idea. How did the water molecules move? Just like the smoke. They diffuse. Does diffusion take energy? No. And this is why the liquid went up. A couple more. Number 5, why did we use distilled water? Let's start off with what is distilled water?
Like almost boil down water.
What do you mean by boil down?
Like it removes. Don't babies use it in their bottles?
I don't know. I've never had any babies.
I think they do, but it like boils off so much. I don't know what it feels like.
Yeah.
Let's say evaporated.
Not for distilled. You guys are tiptoeing around the right idea. What's removed from the water if you distill it? Everything else. Right. So distilled water is pure. It's 100% water. What was the whole point of the lab? To show what? What did the water do? Went in the egg by osmosis. What causes osmosis? If there are equal concentrations, would osmosis occur? Right. So we use 100% water to create the biggest possible concentration difference, which causes osmosis. Osmosis is caused by a concentration difference. By using pure water, we maximize the concentration difference and osmosis, which is what the lab was intended to show you, is osmosis. Last one. What would happen if we forgot to poke a hole in the egg? Would there still be higher concentration in the beaker of water? The membrane, would it still let water in? Yeah, so eventually when the water keeps moving in, the egg will probably crack, right? The straw did not affect the membrane's permeability or concentration difference of water. So water would still move in the egg. And eventually crack it. Your conclusion should be about this length, a few sentences for each one. Hopefully this little discussion gave you some ideas. Are there any that we need to revisit or go back to? Let's talk about your graph then. If you haven't done so already for your graph, you need to open up a Google Sheet. And then the x value, which is the independent variable, is what? What did we change on purpose in the experiment? We exposed the shell. That was part of our setup, but we didn't change it throughout our experiment. What did we change throughout our experiment? We took all the different measurements at different times, right? The time would be the independent variable. So you should have time like 5, 10, 15, 20, etc. And then you should have how much the fluid rose or rise. And that was in millimeters. And I'm just going to make some numbers up. We'll say 10, and then it went up another 15, then 40, and 50. If you put letters with your numbers, it won't graph. So if you put your units in with the 5 and the 10 and the 15, your graph won't work. So just put your units up top. Then you select, hit the chart button here. And what type of graph should we use? Bar is for categories. Or is time all those different categories? Nope. A line graph shows continuous change. So was the time continuously running? Yeah. So we want to use a line graph. We have nothing on the x-axis. So here our time, the blue line, is a line. We want it as the x-axis. So to switch that, you go to Chart Types, and you click this little box, Use Column A as Labels. And then to add a better title and label the axes, you just go under Customization and find, you know, the title section and the axes section. And then you hit Insert and eventually copy it into your lab report. I think you should be set up pretty well to write a good lab report and finish it up. We'll probably be working on it today and tomorrow. So at this point, you can get back in your group and start cranking out the conclusion question. Number 1 on the extension, you need a little previous knowledge on this. If we used hot water, what do you think would change in our experiments? Any ideas? We know the lab was about particles, water in particular, spreading out or diffusing across the membrane. And we know that diffusion is caused by the random motion of particles. How does heat influence or connect to one of those ideas?
Would it just go by faster?
Yes, it would. Why? Right. If you heat a substance, the particles would be moving faster, so we would expect the liquid in the straw to rise up quicker. Good. 2A, what types of molecules are in egg cells? We've studied these extensively already this year. Proteins, lipids, and some carbs. Are those molecules found in actual living cells? Yes. So our model is pretty good in that sense. How are the membranes similar and different? We talked about this in our opening question. Do both membranes let anything pass through? Anything? No, they're semi-permeable, right? Both. That didn't work well. I need to calibrate. Both are semi-permeable, which means they allow some particles to pass and others not to pass through. And it turns out, like we showed in the experiment, larger molecules can't pass through the cell's membrane directly either. So you could add that in there. Oh, I just answered the next question. So larger molecules generally can't go directly through the membrane. Bless you. Yes? We're not finished with the story of cell transport yet. They can get through the membrane. There's like a special passage for them. They don't move directly through. There's like a little tunnel called transport proteins. We're going to actually talk about those tomorrow. But I like the way you're thinking now. And your last question asks you to do a little math. Calculate the rate of osmosis using data from your lab. So water moved into the straw in a three-dimensional space. So the first thing you have to do is calculate the volume of water that moved into the straw. So the volume of a cylinder is the height times pi times the radius squared. So you will need to, actually we can just do it together. Grab a straw, and if you measure the radius, we're going to call it 5 millimeters. So use that for your radius value. And then where do I get h from? The height of the cylinder. What would that be? Tarun? The whole straw? Remember, we're calculating the rate of osmosis. So how did we quantify how much liquid went into the straw? Did we just say it was this much? Right, how high the liquid rose. So H will be how much the liquid rose over your entire experiment. That gives us the total volume of osmosis. How would we convert that to a rate? A rate takes into consideration how long it took. Like if you're driving down the road, you could say I'm driving at a rate of 60 miles per hour. So what do we have to do to our volume to convert it to a rate of osmosis? Yes, Will?
Every 5 minutes.
Right, so we're going to divide by time. And whatever value you use for H, right, I would probably use the last one. Make sure you use that corresponding time. You can't use the height, from 5 minutes and then divide by 20 minutes. That's not an accurate representation of how quickly osmosis happens. Are there any of these that we still need to talk about that you're feeling shaky about? Yes?
Do you want centimeters per second or centimeters per minute?
It's going to be millimeters, because that's what we measured in. And you can do minute. That's fine, since we also measured every 5 minutes. I think that'd be the easiest. A couple more things. When you make your graph, we're showing the change in the fluid over time. Your independent variable goes on the x-axis, so what should be on the x-axis? Time, right. That's what we manipulated. We waited and saw how long it took for the fluid to rise. Also, what type of graph should we use? Is it reasonable to draw a line and say that this much osmosis happened between 5 and 10 minutes? If we have 2 data points, would that be a reasonable assumption or not? Absolutely would. Sure. Since they're not separate categories, you can interpolate, which means assume that between your data points, the same pattern occurred. So you want to use a line graph. That's the point.